Good morning everyone. Today we'll discuss about the functions of hemoglobin, metabolism and synthesis of hemoglobin and different porphyrias. So today we will discuss about the competency B16.11 functions of heme metabolism, porphyrias and bile pigments. So the learning objective: identify the two amphibolic intermediate from which heme is synthesized, and at the end of this session everyone should be able to name the key enzyme of hepatic heme biosynthesis. And at the end of this session everyone should be able to explain why porphyrinogens, porphyrins both are tetrapyrols and porphyrins are colored, whereas porphyrin are colorless. Everyone should know the different enzymes of heme biosynthesis. Some are mitochondrial enzymes and some are cytosolic enzymes. So what are the mitochondrial heme synthesis enzyme and what are the cytosolic heme synthesis enzyme? Everyone should know at the end of this session. Everyone should be able to indicate the step conversion of heme to bilirubin in the cytoplasm as well as the steps in the mitochondria. And at the end of this session, everyone should be able to understand and what are the clinical presentation and what are the mechanism of different porphyrias. And at the end of this session, everyone should be able to know how bilirubin is metabolized and what is the definition of jaundice and what are the different type of jaundice. What are the approach for determining its biochemical underpinnings. So again, coming to the biochemical importance, at the end of this session, everyone should be able to know what are the biochemistry of porphyrins and what are the bile pigments and how is heme synthesized, the different steps, and porphyrins and iron. And the biochemistry of porphyrin of heme and the basic for understanding the varied functions of the different hemoprotein. And we also have to understand the importance of porphyrias, which is a group of disease caused by the abnormalities in the pathway of porphyrin biosynthesis. So again, the biochemical importance. Jaundice is very common. So what is jaundice? And it's a much more common clinical condition, a consequence elevated where there is increased level of plasma bilirubin due to either overproduction of bilirubin or due to failure of excretion. So jaundice occurs in numerous diseases ranging from hemolytic anemias to different viral hepatitis to cancer, especially the cancer of head of the pancreas. So now coming to the porphyrins. What are these porphyrins? These porphyrins are cyclic compounds formed by linkage of 4 pyrrole rings through methionine bridge. So this is the structure of pyrrole and structure of porphyrin. So naturally occurring porphyrins, various side chains are replaced by 8 numbered hydrogen atoms in the pyrroles, and so this structure represents the shorthand representation of a porphyrin, uroporphyrin 3. In the left, number 1 through 8 correspond to those, and the right, the substitute of uroporphyrin 3, and A indicate acetate, B indicate propionate, and we can see the asymmetry of the substituents on ring 4. So uroporphyrins were the first bound in the urine, but they are not restricted to urine. And core porphyrins were first isolated from feces, but they are also found in the urine. So uroporphyrin 1, then uroporphyrin 3, core porphyrin 1 and core porphyrin 3. So porphyrin form complexes with metal ions that bind to the nitrogen atom of each of the 4 pyrrole rings. Example, iron porphyrin, hemoglobin, the magnesium containing porphyrin, that is chlorophyll, the photosynthesis pigment in plants. So heme proteins, which are ubiquitous in the biology. This hemoglobin, heme, serve diverse functions including but not limited to the oxygen transport and storage, for example, hemoglobin and myoglobin. And electron transport, for example, cytochrome C and cytochrome P450. Heme are tetrapyrrole, of which the 2 type heme B and heme C predominate. So these are the structure of heme B and heme C. So heme C, the vinyl group, and heme B replaced by covalent thioether links to an apoprotein, typically via cysteine residues. Unlike heme B, heme C does not readily dissociate from each apoprotein. So vertebrate hollow protein generally bind 1 molecule of heme per mole, although those non-vertebrate may bind significantly more heme. So these are some examples of some important human and animal hemoproteins. The protein hemoglobin, the function is transport of oxygen in the blood, myoglobin, storage of oxygen in the muscle, and cytochrome C involved in the electron transport chain, and cytochrome P450 in the hydroxylation of xenobiotics, and catalase degradation of hydrogen peroxide, and tryptophan pyrrolase oxidation of tryptophan. So the functions of the upper protein we will be discussing in further slides. So heme synthesis. Heme is synthesized from succinyl-CoA and glycine. Heme biosynthesis occurs in most mammalian cells except mature erythrocyte, which lacks mitochondria. So except mature erythrocyte, please remember, and where there is no mitochondria. So 85% of heme synthesis occur in erythroid precursor cells, that is in the bone marrow, and the majority of the remainder in hepatocytes. So heme biosynthesis is initiated by the condensation of succinyl-CoA and glycine, which is a pyridoxal phosphate dependent reaction catalyzed by mitochondrial enzyme, that is the delta amino levulonate synthase, ALA synthase, which is also an enzyme of rate limiting step of heme biosynthesis. So the reaction here, succinyl-CoA plus glycine, it will form delta amino levulonate, coenzyme S8, and carbon dioxide will be released. So human express two isozymes of ALA synthase 1 ubiquitously expressed throughout the body, whereas ALA synthase 2 expressed erythrocyte precursor cells. So the reaction, which is rate limiting for porphyrin biosynthesis in the mammalian liver, initially product is formed, that is alpha amino and beta keto adipate, which is rapidly decarboxylated to delta amino levulate. So the succinyl-CoA, the active succinate, and the glycine will condense, and it will be reacted by the enzyme ALA synthase, and the pyridoxal phosphate is the coenzyme here. It will form alpha amino B keto adipate, and then further reaction, which is catalyzed by ALA synthase, and carbon dioxide will be released. So it will form delta amino levulonate, and further 2 molecules of delta amino levulonate will condense, and water will be released by the enzyme ALA dehydratase, and finally the pro-porphyrinogen, the first precursor of pyrrole, is formed. So this is the reactions where biosynthesis of pro-porphyrinogen is the first precursor pyrrole is formed, and ALA synthase is present in the mitochondria, and whereas ALA dehydratase is present in the cytosol. So the reaction takes place both in the mitochondria and in the cytosol. Porphyrin and ALA synthase is present in the mitochondria, and ALA dehydratase, as I mentioned, it is present in the cytosol. So condensation of 2 molecular ALA catalyzed by cytosolic ALA dehydratase form porphobilinogen. So the second reaction here, 2 molecule of delta amino levulonate, then porphobilinogen, and water is released. So a zinc metalloprotein ALA dehydratase, which is sensitive to inhibition by lead, which occur during the lead poisoning. So reaction is catalyzed by cytosolic hydroxymethylabine synthase and uroporphyrinogen 1 synthase, which forms hydroxymethylabelin. 4-porphyrin reacts with water and forms hydroxymethylabelin and 4 molecules of ammonia. And this reaction, the catalysis involves head-to-tail condensation of 4 molecules of 4-porphyrin to form the linear tetrapyrrole, that is hydroxymethylvaline. And further, there will be cyclization of hydroxymethylvalase, which is catalyzed by cytosolic uroporphyrinogen 3 synthase. And in the third reaction, this hydroxymethylvaline will be converted to uroporphyrinogen 3 and water. And this formation of uroporphyrinogen 3, then hydroxybilane, can also be cyclized spontaneously to form uroporphyrinogen unconditional condition. Uroporphyrinogen is formed exclusively the type 3 isomer, formed in almost exclusively the type 3 isomer. The type 1 isomer of porphyrinogens are however formed in excess in certain porphyrias, since the pyrrole ring of this uroporphyrinogen are connected by methionine rather than methane bridges. They do not form a conjugated ring system, and thus they are, and indeed all porphyrinogens are colorless. So they are, however, readily auto-oxidized to their respective colored porphyrins. So all four acetate moieties of uroporphyrinogen 3 next undergo decarboxylation to methyl M substituents, forming core porphyrinogen 3 in a cytosiltic reaction catalyzed by uroporphyrinogen decarboxylase. So this is the reaction how uroporphyrinogen 3 is converted to core porphyrinogen 3 by the enzyme uroporphyrinogen decarboxylase, and 4 molecule of carbon dioxide is released. So A stands for acetyl, M stands for methyl, and P stands for propionyl. In the fifth reaction, uroporphyrinogen 3 to core porphyrinogen 3 and release of 4 molecule of carbon dioxide. So decarboxylate can also convert uroporphyrinogen 1, which is a mitochondrial enzyme. Then core porphyrinogen 3 enters mitochondria, converted to protoporphyrinogen 3. Then core porphyrinogen oxidase enzyme decarboxylate further oxidizes the 2 propionic acid side chain to form protoporphyrinogen 3. In the sixth reaction, this core porphyrinogen 3 with the molecular oxygen and hydrogen, protoporphyrinogen 3 is formed, and there is release of 2 molecule of carbon dioxide and 2 molecule of water. The enzyme oxidase, which is specific for type 3 core porphyrinogen and type 1 protoporphyrin, do not occur in humans. Protoporphyrinogen 3 net oxidized results protoporphyrin 3, the reaction catalyzed by protoporphyrinogen oxidase. The seventh reaction, protoporphyrin 3 plus 3 molecule of oxygen results protoporphyrin 3 and 3 molecule of hydrogen peroxide. So the final step of heme synthesis involves the incorporation of ferrous iron to protoporphyrin 3, and the reaction is catalyzed by the enzyme ferrochelatase. And finally, protoporphyrinogen 3 plus iron, so heme is formed. So this is the summary, step and cellular location of the reaction in the biosynthesis from porphyrinogen of the induced porphyrin derivative, notably heme. So this is how conversion of porphyrinogen to uroporphyrinogen take place. Linearization of porphyrinogen is catalyzed by hydroxymethylvaline synthase, also called uroporphyrinogen synthase 1 or porphyrinogen deaminase. So core porphyrinogen 3 then enters into the mitochondria, where it is converted to protoporphyrin 3. So several steps are involved in this conversion. The mitochondrial enzyme core porphyrin oxidase catalyzes the decarboxylation and further oxidation of the 2 propionic side chain to form protoporphyrin. The enzymes are able to act only on type 3 core porphyrin, which would explain why type 1 porphyrins do not generally occur in nature. And further oxidation of this protoporphyrin to protoporphyrin, which is catalyzed by another mitochondrial enzyme, that is protoporphyrin oxidase. In mammalian liver, the conversion of this coporphyrin to porphyrin requires the molecular oxygen.